What I'd like to do in this video is briefly go over the last two questions because we didn't get a full chance to talk about them in any real detail in class. So question 5 on the homework for chapter 5 was asking about how we can use these neuropharmacological tools that are shown in figure 6-7 to try to decide for a case where we have a couple synapses what kind of a particular acetylcholine receptor is found in those situations. So let me flip over to the figure. The key thing here, and this is what we were talking about at the end of the class, is that we have these different kinds of drugs that we can use that can stimulate acetylcholine receptors or inhibit them. So the idea is that there's two different kinds of nicotinic acetylcholine receptors. There are nicotinic receptors, and there are also muscarinic receptors. Both of them will bind acetylcholine, and they're both found in different synapses around our bodies. In the neuromuscular junction, for example, we have lots of nicotinic receptors, which becomes important when we talk about myasthenia gravis in the next question. It's difficult to distinguish them just by playing around with acetylcholine, though. If we give acetylcholine to a tissue that has nicotinic receptors, it'll be responsive, as will a tissue that has muscarinic receptors. So we really need additional tools, and that's where these other drugs come in. So in the case of nicotinic receptors, we have a drug nicotine that is the same nicotine found in tobacco, where its effect is that it directly binds to these nicotinic receptors in the same way that acetylcholine does, and it stimulates them. So we call nicotine an agonist. Similar, or in contrast to that, we have another compound called curare, which is an antagonist that functions in the opposite way. It will also bind specifically to nicotinic receptors, but when it does so, it will block the nicotinic receptor, and certainly it won't open the nicotinic channel, but it'll also prevent any acetylcholine or nicotine or any other kind of compound from binding to that receptor and stimulating it. So we have a blocker, an antagonist, and we have an agonist, nicotine, and both of them are very specific to the nicotinic receptor. Neither of them will bind to the other kind of acetylcholine receptor, the muscarinic receptor. And in the case of the muscarinic receptor, we fortunately also have drugs. We have both an agonist, which is muscarine, and atropine, which is an antagonist, that also interact in the same way with a muscarinic receptor as nicotine and curare act on the nicotinic receptor. So if you're interested in a particular synapse and you're trying to figure out what kind of acetylcholine receptor is found there, and you know the thing is present, or that synapse has let's start over. We know the synapse in question has acetylcholine receptors, but we don't know what kind of receptor is there. We could employ these neuropharmacological tools in order to answer the question. So an experiment we could do, if the hypothesis is, for example, that a particular synapse has nicotinic receptors and not muscarinic receptors, we could dose that synapse with nicotine, which should stimulate nicotinic receptors, and at the same time we could also dose it with atropine, which should inhibit any muscarinic receptors that are that could be present. If we're right that the synapse has nicotinic receptors, that should cause those receptors to be stimulated. Then we'd want to do the opposite experiment, where we give that same synapse muscarine, which should stimulate muscarinic receptors but not nicotinic receptors. And we probably also would want to give that same synapse curare to block the nicotinic receptors. And in that situation, we should see no stimulation of the synapse. And we could repeat that in multiple synapses. So in the question, it asks about a parasympathetic pathway that has two synapses in a row. One synapse is between one neuron and another neuron. And in that case, what I will tell you, if we went through that series of experiments, what we'd find is that when we exposed it to nicotine and atropine, we'd have very strong stimulation of that first synapse. But if we exposed it to curare and muscarine, we would get no response because those synapses do not have muscarinic receptors. They just have nicotinic receptors. In contrast, if we followed that to the end, say we found some heart tissue, like we talked about in the first question in this chapter, the heart tissue uses acetylcholine, but it has muscarinic receptors. And so in that case, if we stimulated the heart tissue with nicotine, which does not bind to muscarinic receptors, and atropine, which blocks muscarinic receptors, we should get no response. We would not get a slowing of the heart rate in that case. But if we dosed that same tissue to muscarine, and then we exposed it to curare, we would get a nice strong response of the heart tissue because that has muscarinic receptors. So that's what I wrote in my recap here, and if you want, you can pause this, but I'll read through it pretty quickly. Neuropharmacology provides us with tools that we can use to either stimulate or inhibit a neurotransmitter's receptor without using the neurotransmitter. An agonist will bind to the receptor and stimulate it as if it were the neurotransmitter, and an antagonist will bind to but then block the receptor, preventing it from being stimulated by other compounds. Acetylcholine has two kinds of receptors. One is a nicotinic receptor. It's a channel link receptor. The other, muscarinic receptor, is a G protein coupled receptor. That harkens back to earlier slides from chapter 5. Those two receptors are named for their agonist. Each specific kind of receptor has a specific antagonist too. Curare blocks the nicotinic receptor, while atropine blocks the muscarinic receptor. An experiment that we could do would be to deliver nicotine and the muscarinic blocker atropine to each of the two synapses in question and measure whether or not there are responses. We could then deliver muscarine and the nicotinic blocker curare to both synapses and measure the response. The prediction is that a nicotinic receptor synapse only would work when we deliver both nicotine and atropine. But in contrast, muscarinic receptors only would work when we deliver both muscarine and curare. All right, so then that's question 5. Let's scroll down to 6. So question 6 is asking about a disorder. It's an autoimmune disorder called myasthenia gravis that occurs and does directly influence synaptic transmission. So the first, we'll go through these sort of in sequence. So the first question is, what are the disease's symptoms? And when you see a patient with myasthenia gravis, they are characterized by having a substantial loss in muscle tone and ability to contract their muscles in general. And that's particularly true in muscles of the face and the extremities. You'll see, for example, that their eyes will, the skin around their eyes and their eyelids will tend to start to droop because they just don't have the muscle ability to get their eyes all the way wide open again. They also will start to complain, if you talk to them, about focus issues with their eyes because some of the muscles within their eyes are not working very well. They also lose strength out in their extremities, so their muscles that control their hands, for example, may become very weak. And these can be sort of cyclical symptoms that'll phase in and phase out. It's caused when, as I mentioned, it's an autoimmune disorder, so it's caused when a person is generating autoantibodies that attack their acetylcholine receptors, and specifically they're attacking the nicotinic acetylcholine receptors that are found at the neuromuscular junction. In this case, what happens is because the antibodies are binding to their acetylcholine receptors, that causes those receptors to become blocked, and often ultimately causes those receptors to get reabsorbed by the cell, and that results in fewer available functional receptors for acetylcholine in the neuromuscular junctions. As a result, even though the motor neurons are still releasing acetylcholine, the skeletal muscle no longer is receiving its normal level of stimulation to contract. So if I flip over just real quickly to a slide from lecture, so this was a depiction of a synapse. Basically what's happening here is we have a problem on these receptors that are scattered along the postsynaptic cell, and in this case it would be a skeletal muscle cell, where we're still releasing neurotransmitter, but the receptors are being blocked and destroyed, and as a result of that destruction, there are fewer receptors down here to bind the same amount of neurotransmitter, which means that the skeletal muscle cell, the postsynaptic cell, is no longer going to be responsive to the neurotransmitter that's receiving. Now, the last question asks, why might we use acetylcholinesterase? And the key thing that acetylcholinesterase does for us is that acetylcholinesterase is an enzyme that destroys acetylcholine. It was discussed on this slide right here. And so the idea is that this enzyme, what its function is to break down acetylcholine after it's been released by the presynaptic cell. In this case, what we do is we deliver an acetylcholine esterase inhibitor, which would block that particular enzyme and prevent it from breaking down acetylcholine at the normal rate. And so as a result, acetylcholine will linger in that synapse within the neuromuscular junction and therefore provide longer term and better stimulation of any remaining receptors that do happen to still be there and haven't been destroyed by the autoimmune disorder. So again, here is that same statement. So by treating with an acetylcholinesterase inhibitor, we prevent the breakdown of acetylcholine in the neuromuscular junction, and this causes it to linger in that synapse, which increases the stimulation of any acetylcholine receptors that remain in the synapse and therefore increases the responses of the muscle cells. This is actually sometimes done as a diagnostic test to verify that somebody has myasthenia gravis. They'll give them an acetylcholinesterase inhibitor, and all of a sudden a person's muscle functions will sort of snap back to normal. And that's a pretty good indication then that the issue here is this loss of receptors. Unfortunately, the problem is you can't consistently treat with an acetylcholinesterase inhibitor over long term. Ultimately, what will happen is you will have some kind of an acclimation effect that causes the drug to no longer have an effect after even just a fairly short period of time of dosing. But it can be used at least for short-term relief from the symptoms. I hope that helps with those last two questions. They are part of the exam, and I think that you did get to talk about them in groups, and it didn't sound like there was a lot of confusion on either point. If you do have confusion, though, please consider talking to me at the review session on Thursday, or send me an email, or stop by my office. I'm happy to talk to you more about these questions. Thank you very much. Hope that was helpful.